Hello everyone. Today I am going to explain one experiment which is one of the common experiment of fermentation technology or bioprocess engineering. This is optimization of media parameters as well as environmental parameter for growth and product formation. Say for example, today I am going to explain about the optimization of conditions such as pH, temperature, carbon source and nitrogen source for the production of biomass as well as amylase production. See, for this experiment, we require one microorganism. Since our product is amylase, we can choose any one amylase producer. Say for example, we have chosen Bacillus subtilis. Now our target is to improve the amount of amylase production using Bacillus subtilis by providing optimum condition. See, we know very well that microbial product can be improved by providing optimum conditions as well as by changing certain specific media parameters, and this is called optimization. So our aim is to optimize proper environmental conditions such as pH and temperature, as well as we are going to change the media composition. Since we can use several type of media compositions, but for your experimental point of view, I have taken only four parameters: two environmental parameter, that is pH and temperature, and two media composition, carbon and nitrogen source. But in real experiment, you are having n number of experiment, and you know very well that we are having two type of approach. One is statistical, other is non-statistical approach. By using a statistical approach or placket Berman design, you can reduce the number of parameter and you can proceed with the further surface response methodology. But here our aim is to explain by traditional method, that is one factor at a time method. So our method is non-statistical. OFAT, it means one factor at a time method. If we will take more and more parameters, in that case the work is very tedious. Since this is just for the learning purpose or experimental purpose, we are taking only four parameters and we are going through traditional classical approach, that is non-statistical one factor at a time method. At first you can choose microorganism, you can isolate microorganism from the environment. Say for example, we have already isolated Bacillus subtilis, which are able to produce amylase. Now our aim is to optimize the media in which Bacillus subtilis is able to grow. At first, we can choose those media which is already available for the growth of Bacillus subtilis as well as production of Bacillus subtilis. We are having two ideas. Number one, you are having separate parameters for the growth and separate parameter for the product formation. Or second idea is you can fix the conditions of temperature, pH as well as media. Everything is fixed, and in that particular condition you want to see that the bacteria is growing with this particular rate and what is the exact amount of product formation. For your convenience, we have taken fixed parameter, fixed temperature, fixed pH, fixed carbon source, fixed nitrogen source, which is already available in your database or in research paper. In this particular condition, this Bacillus subtilis is showing good amount of growth as well as product formation. But now for our own experiment, for our own bacteria, we are going to optimize the conditions as well as optimum media parameters. So what you will do for here, for the amylase production, you can change different pH and temperature. Whatever the set pH and temperature is there, you can shuffle a little bit more or less, and then you can also change different carbon sources as well as nitrogen sources. See, optimization is important for various reasons, but most important reason is you want to improve the product formation. So here at first I have taken optimization of pH. As you know, pH is very important parameter for the growth and product formation. At first you can choose one common basal media, and in this media parameter, as I already said, you can set a fixed condition such as here I am providing starch as a carbon source, then yeast extract, peptone as a nitrogen source, and other salts. Then you can set pH 7 and make up the volume of distilled water 100 ml. So this is the fixed media. Now, if you want to optimize pH by OFAT method, that is one factor at a time method, in this method you can fix all the parameters. You are only going to vary one condition. Since you are going to optimize pH, you can just change pH. Rest all the condition will be same. Here I have taken 5 different pH 5, 6, 7, 8 and 9. So you can make 5 flasks. In all these 5 flasks you can set the same starch, yeast extract, peptone, then salts and distilled water volume, everything is same. Only thing is in this 5 different flasks you are having various pH. In one flask pH is 5, other is 6, then 7, then 8 and finally 9. Now you can start your experiment. You can just pour Bacillus subtilis, active culture of Bacillus subtilis, and you can start your work. After addition of Bacillus subtilis, the bacteria will grow. So what you can do here is you want to measure the growth as well as product formation. First I am going to explain about the growth of bacteria, which can be observed simply by turbidostat method. You can measure the turbidity at using OD 600 nanometer or 660 nanometer. Here I have taken 660 nanometer OD for the measurement of growth of Bacillus subtilis. Later on, we will follow the production, that is amylase production, by using amylase activity, that I will explain later on. At first, what you will do, whatever the 5 flasks you have taken, from this 5 flasks you can obtain certain blank media which is acting as a blank. Then you can add culture. Once you will add culture, you have to measure how much culture you have added. You can measure it simply by taking OD. You can take certain amount of media like 3 ml media, and then you can take OD. Now you can adjust the OD of culture and OD of blank. You can once decrease it, once you can deduct it, you can finally get the value of OD. It means optical density of a particular Bacillus subtilis. Then what you will do is you can take next parameter such as temperature. Here also I have taken the same composition, same starch extract, peptone, salt, pH, but here since want to change the temperature, I have taken 3 temperature. You can take more than that according to your convenience. Here I have taken 3, that is 25, 37 as well as 50 degrees Celsius temperature. Since I know my bacteria, Bacillus subtilis, they are able to grow at high temperature in fact, and hence I have taken up to 50 degrees Celsius temperature. Then I inoculate the microorganism, that is Bacillus subtilis, and repeat the experiment which I have done just before, that is with the pH. It means this 3, for this 3 flask you can take out the media, then you can add culture, Bacillus subtilis, mix it, shake it well, again obtain culture, take OD of blank media as well as culture, write down the final OD value of the culture, and then you can keep the culture for the incubation period so that when microbes will be grow, their optical density will be increased, their growth will be increased, and simultaneously their product formation will also is going to be increased. Same we have done with the pH. Now third parameter, suppose I have taken nitrogen sources. Here in nitrogen source again every media is same constant, PS7, starch is fixed 0.5%. Only thing is I am going to vary one parameter. Since there are two nitrogen source, one I fixed yeast extract, but other I am going to vary. If you want you can vary two also, but since it's OFET method you are going to vary only one at a time. I am going to vary peptone. Instead of peptone, you can take urea, glycine as well as ammonium sulfate. And you can see in these 4 different flasks which is important for the growth as well as product formation. The procedure is same. You can take the blank, you can add culture, and then you can take OD, and then you can proceed with the further amylase activity. Then last one is carbon source. Here also you can take the same media. In this media composition all the parameters are same, only I am going to vary with the starch. Instead of variation of starch with the other carbon source, I am fixing starch, but I am changing the percentage. So suppose I have taken instead of zero point 0.5 I have taken 0.3, 0.4, 0.5, zero point 0.6 and zero point 0.7. So I have chosen 5 flasks. In this 5 flasks the bacteria has been inoculated. Before inoculation we can take out the media for blank reading, then you can take the culture reading, again same OD value you can get. So you have 4 parameters. In all these 4 parameter, what you have done, you have taken certain amount of media, like 3 ml media you have obtained, you have added culture. After addition of culture you can shake it well, again obtain culture, say for example 3 ml culture. So you have 3 ml media, 3 ml culture, and you can take OD of both. By the taking of OD, you are able to get the biomass. So you are knowing now that what is the biomass of your microorganism when you have just inoculated Microbacillus subtilis. Then all these different type of flasks for the carbon, nitrogen, pH and temperature can be kept in incubator in the proper shaker. Suppose I am providing different temperatures, but except the temperature variable, rest all the variables are kept at 37. Then you can keep it for the end time period. Say for example, for your college purpose, suppose you don't have enough time for work continuously, then you can take reading for the 4 hour, 0 hour to 4 hour and 4 hour to 6 hour. You can take any reading. Suppose if you have time, you can take reading of half of hour or 1 hour. After every 1 hour you can take out the media, you can take you can read the OD, and you can proceed with the enzyme activity. But here I have taken only 3 reading, one for 0 hour, other for 4th hour, and third for 6th hour. At every time you can take out the media, that is culture, and from this culture you have to obtain certain amount of culture, take OD and keep it back in the incubation condition. After the 6 hour, what you will see is, suppose I am showing here one example of pH. I have taken 5 pH, that is 5, 6, 7, 8 and 9. In all this, I have already gone through the OD 600. It means I am able to get OD of OD, that is biomass bacillus of Bacillus subtilis, in 0 hour, then 4 hour as well as 6th hour. So by the comparison of this data, I am able to know that which pH is better. Say for example, in this 3 different conditions 0, 4 and 6 hour, we know very well that we are going to use OD method. In this OD method, the total OD cannot be decreased as it is turbidostat. It is based on turbidity. The turbidity can never be decreased. If microbes are inactivated or die still present in the same media, it means turbidity is never going to be decreased. Turbidity will be either increased or it will be constant. It means we know very well that if you are taking 3 different type of different timing, it means maximum OD you will get in 6th hour only. So at 6th hour you can get OD 600. So you can check the OD of 6 hour only, and you can compare of all this 5, 6, 7, 8 and 9, whichever is best you can write down. Say for example you are getting maximum OD at pH 7. It means you'll just note it down that the pH 7 is best condition, that is best pH for the optimum growth of microorganism. Like in the same way, suppose if I am saying that for your experimental condition pH 8 is best and you are getting maximum OD at pH 8, you can write down or note down at pH 8. So you can just see the 6 hour value and you can get the value of pH. But the only problem is, up to this time you have just you are just knowing about the biomass, you don't know about the enzyme activity. Our target was two: number one, optimization of various parameter for maximum growth and maximum production. Growth you observe by taking OD, but maximum production can be observed by enzyme activity. Here I have gone through amylase enzyme. It means I am looking for enzyme production, amylase enzyme production. So I will do enzyme activity for amylase production. So we can proceed further with the amylase activity, enzyme activity. What are the other requirements? You can see you require cell-free extract, starch, phosphate buffer, dinitro-salicylic acid, that is DNSA, and standard solution of maltose. Besides that, you have distilled water, starch, DNSA, buffer, as well as enzyme culture broth. These things you should be ready. How you will get enzyme culture broth? Let me explain. See, when you started your experiment, you have taken different flasks. In this flask, you have just added culture. After addition of culture, you took out the culture and you have taken OD. What you will do is, after taking OD, you can just centrifuge. After centrifugation, the microbial palette will be separated and supernatant will be separated. Since amylase is extracellular enzyme, it means we are 100% sure that this amylase is not present inside the microbe, it is present outside the microbes, it means it is present in media. So what you will do is, after centrifugation of that fermentation broth, you can separate pellet as well as supernatant, and you can check the amylase activity of supernatant only, because your amylase is present in supernatant. Same activity you can repeat for 0 hour, 4 hour, as well as 6 hour. In all these 3 period, you have to take the culture, you can centrifuge, and you can separate the supernatant, and you can work out with the supernatant only amylase activity of the supernatant. In the supernatant, enzyme is present, and that's why I have written here enzyme culture broth. So now I'll explain one stepwise how enzyme activity can be obtained. In this procedure, first you have taken flask, you can take out culture in the different test tubes, and then you can take OD. This is for biomass. Then with this culture only, you can go for centrifugation. Once you will centrifuge it properly, one is blank and other culture. Once you centrifuge it properly, you can get supernatant as well as pellet. Now you can remove supernatant and you can proceed with the enzyme activity, that is amylase activity, which is present in supernatant. So what you will do, that I will explain, but before that you have to see your target. Your target is, if you want to proceed with the amylase activity, you have to take one blank and two test culture, two test tubes. So you can take three test tube, one is for blank and two test tube, write down test 1 and test 2. In blank you can add 1 ml buffer, in the test you can add 0.5 ml buffer. Then you can add supernatant in which enzymes are present, 0.5 ml. Then you can use starch as a substrate and incubate for 37 degrees Celsius for 10 minutes. In this 10 minutes, your enzyme will hydrolyze your substrate, that is starch. Then you want to measure the amount of maltose produced. For that you can add densa. Then after addition of 1 ml of densa, you can keep it at 100 degrees Celsius in water bath. The color will be dark, and then you can minimize the color by addition of distilled water and check the OD at 540 nanometer. See, this is a common experiment which you have done in your college, but still here I have already explained. What you will do is, first you can take a test tube, you can add buffer, starch, and enzyme. So one by one you have to add in the blank as well as in other one. Suppose I have added buffer, then starch, and then you can add enzymes. So one by one you have to add in all blank test 1 as well as test 2. It means you have 3 tubes. Now you can incubate at 37 degrees Celsius for 10 minutes. Now your enzyme will break down substrate, that is starch, and hence whatever the product is formed, that product can be observed, but for observation of your product you have to add DNSA reagent. Suppose you added, after addition the color will be changed, but the same will be repeat for all the tests. But once you will keep it in the boiling water bath, in the boiling water bath the color becomes darker. Then you can add distilled water so that the color will be a little bit minimized. and then you can take OD at 540 nanometer, which I have explained in the previous observation table. Now you have only OD at 540 nanometer. Now you have to go through enzyme activity or amylase activity. For that, you know very well that enzyme activity is nothing, it's just a measurement of one unit. So we have to go through unit activity of enzyme. It is the amount of enzyme that catalyzes the reaction of 1 micromole of substrate per ml per minute under standard condition. So how you will do this? For that you require standard graph of maltose that you can prepare separately using DNSA method, and you can get the slope value of maltose. Now what you will do? For the enzyme activity, the formula is already known. It is concentration of maltose upon molecular weight of maltose, then into 1 by incubation time into 1 by aliquot of enzymes. See here, your target is already fixed. You want to go for enzyme activity, only you have to focus on concentration of maltose, because molecular weight of maltose is 342, that is well known. Incubation time you have already given 10 minutes, and aliquot of enzyme it was also fixed 0.5 ml. If by using this graph, you are able to obtain the value of concentration of maltose. I just repeat. See, you already get the value of 540 nanometer, OD 540 nanometer. By getting OD 540 nanometer, you can calculate concentration. You have already graph of OD versus concentration. Using standard graph of OD versus concentration, you can get value of concentration of unknown maltose. Again I repeat, by using standard graph of maltose, you can get the value of unknown concentration of maltose, which you get from this flask. And then you can calculate and you can get final enzyme activity. So our target is to focus on concentration of maltose, and whatever the activity we will get, the unit will be micromole per ml per minute, or it is also simply considered as unit value. If you will make graph for this, you are having this observation table. You can see, suppose I'm using the same, that is pH. Biomass you have already obtained, and you will get now the value of enzyme activity. So now your table is completed. But the problem is, see, enzyme activity you have to compare from 0 hour to 4 hour to 6 hour, and you have to see each and every value, and hence you have to prepare graph so that you can easily see in which proper pH the maximum enzyme activity is present. For the graph you can take two axes. One, in the x-axis you can write times in hour, and in the y-axis you can write enzyme activity in terms of unit. Then you can mention your timing. Suppose my timing is 0 hour, 4 hour and 6 hour. Then I have to prepare graph. See, is all this 5 value which I have taken, pH 4, 3, 4, 5, 6, 7, 8, 9. I have just plotted the graph from 0 hour to 4 hour, how much amylase production was there. That is through enzyme activity. And then all this where what is the position of this all enzyme activity at 6 hour. And you can see among all these 5 parameter in this graph, it is showing that pH 7 is having highest enzyme activity. So I can say simply that the pH 7 is the best condition, that is best pH at which optimum amylase activity can be possible. This is your result. Like in the same way, you can practice for other parameters. For your practice, I have already given one value, and you can go through this practice. Say for example, for pH 5, I have already given biomass as well as OD 540 nanometer. For biomass, you can simply see that which biomass is best, but for the enzyme activity, you have to go through the data of OD 540 nanometer. So for your practice, I have given OD at 540 nanometer. You can calculate enzyme activity using the formula which you know very well. One thing you need, that is maltose, standard graph of maltose. You might have done this experiment, standard graph of maltose, earlier in your early classes. You can use that standard graph and calculate enzyme activity. Like in the same way, you have given other data, that is for the carbon sources. You can go through this carbon sources, calculate enzyme activity value. Then like in the same way, for the nitrogen sources, I have given all the value. You have to calculate enzyme activity, as well as I have given one value of temperature. This is for your practice. Whenever you are doing your work, you will get the same type of table, and you can calculate enzyme activity, and you have to plot all the four different graphs, as I have already mentioned earlier. Like in the same way, you can prepare all the four different graphs. Hope you understand. See you.